Assalamualaikum warahmatullahi wabarakatuh. I bid to Dr. Nadia and my fellow friends. So today we will going to present to you the topic 60, which are nutrients, translocation and hormone implants in subtopic transport mechanism in plants. Me, Nur Nadira binti Saiful, AS10838 will present to you the vascular tissues. My friends Ellie will present about the water transport on xylem, and Izati will present organic nutrient transport in flower. So we'll be proceed to the next slide. Alright, vascular tissues. Vascular tissues consist of two types, which are xylem and phloem. The roles of xylems are to transport water and minerals to the leaves in the plant, while the roles of phloems are to transport the organic minerals to the leaf in the plants. Okay, we proceed to the next slide. Okay, next is about the plant transports and the water potential. Okay, we must know that potential energy is a stored energy. Okay, the water potential is the energy of the water. So we know that water moves passively by using the concentration gradient, which are from the higher potential to a region of the lower potential. There are two factors that determine the water potential in terms of the cell, which are the water pressure across the membrane and the solute concentration across the membrane. Okay, this is the phloem structure. Okay, the conducting cells in this phloem structure is the sieve tube members. Okay, other than that, they also have the companion cells that provide the proteins, and then the end walls of the sieve tube elements ended with sieve plate, and the plasmodesmata are extended to the sieve plates. So we proceed to the next slide. Okay, next is about the plant transports and the water potential. Okay, we must know that potential energy is a stored energy. Okay, the water potential is the energy of the water. So we know that water moves passively by using the concentration gradient, which are from the higher potential to a region of the lower potential. There are two factors that determine the water potential in terms of the cell, which are the water pressure across the membrane and the solute concentration across the membrane. Alright, what is the concept of the water potential? Okay, the concept of the water potential are divided into two, which are the pressure potential and the osmotic potential. The pressure potential is the effect of the pressure that has on the water potential. So, pressure. So we know that the water will move across the membrane from the area of the higher pressure to the lower pressure. So the hypothesis are the higher the water pressure, the higher the water potential. Other than that, we must know that trigger pressure are the pressure potential that increase due to the osmosis. Okay, the next point are the osmotic potential. The osmotic potential takes into the account because of the presence of the solutes like the salt, sugar, and other solutes. Okay, the water tends to move from the area of the lower solute concentration to the area of the higher solute concentration. So the hypothesis are the lower the concentration of the solutes, which are the osmotic potential, the higher the water potential. It can be seen from this picture. Alright. So these are the examples of the water potential electrical pressure. Okay, here we can see the plant cells need water. And here we can see the plant cells are turgid. Okay, the plant cells need water because of the extracellular fluid are higher in water potential, pressure potential, osmotic potential. This is big when the outside of the outside fluid are higher, so the water inside the cell will tend to move out because of the inside of the cell are lower in the water potential, pressure potential, and osmotic potential. Because the water moves out from the cell, which are to the higher pressure potential, the water will be willed. Okay, but when the water enters the cell, the water potential inside and the outside of the cell are equal, which are no water movements. Alright. Other than that, the pressure potential will increase until the cells are turgid, which it cannot accept more water. It cannot accept more water, or it accept excess water until it became swallowed. Alright.
Thank you, Nadira, for your explanation. I will continue on by explaining the water transport in xylem. So, basically, the water transport in xylem is mainly influenced by transpiration process. And transpiration process consists of many things that I will be explaining later on. So first, I will explain how water enters the plant. Water enters the plants through the roots, as stated here. Water enters the root by osmosis, where the high concentration of water enters the low concentration of water place, which is the roots, and through osmosis, a new pressure will form, which is root pressure, and this root pressure will push the water in the roots up a bit until the stem, where it will be carried by the transpiration force. It is also speculated that the root pressure is the cause of guttation. Guttation is when the water droplets form at the edges of the leaves. So, this diagram here also shows a brief process of transpiration and root pressure. So, at the stem, after the water molecules are pushed by root pressure, it is pulled up by a process called transpiration. Transpiration consists of two things, which first is cohesion and adhesion force at the stem, and the evaporation process when the water molecules leave stoma. It's also stated that 90% of water is lost during all of this process. Transpiration also relies heavily on stomates being open. So, what is cohesion and tension, or cohesion and adhesion? Cohesion is the tendency of the water molecules right here to cling to each other, as shown here. Whereby adhesion is when is the ability of the water molecules to stick to the xylem cell wall, like shown here. All of these are taken at the plant stem. This is the plant stem, the xylem cell wall at the plant stem. So as you can see, it is a passively continuous upward flow. This is believed to be due to the transpiration. All of this water will be flown to the leaves. I will continue on explaining the cohesion tension model of xylem transport. I will start on the roots, where at the roots the water enters the xylem passively by osmosis, as I have explained earlier, and is pulled outwards due to the tension of the xylem. As you can see in the photo, the waters from the soil enters the xylem at the root. This is the root, and this is the xylem. And the water, and this water column of xylem here, it's from here, extends from here to the leaf. Next, after the water is absorbed at roots, the tension in the xylem will pull the water molecules upwards. As I explained briefly earlier, the water will be pulled by two forces, cohesion and adhesion. The cohesion is where the hydrogen bonding between water molecules are pulled together, and an adhesion when the water molecules are sticking to the wall, cell wall. So all of this, basically the function of cohesion force, it ensures that the water is continuous, and adhesion ensures that the water is in place. It's not everywhere. It's in the straight line moving up. Thirdly, after the stem, the water will flow to the leaf, where the stomata is located. So at here is where the desperation process occurs. When the water will leave through the stomata and will be evaporated at the surface of the leaf. The desperation here is what creates the tension of the xylem that can be located back to the root. So, as I said earlier, 90% of water are lost during this process, but most of them are continuously being replaced by the water absorbed by the water absorbed by the replaced by the waters from the leaves. So the plants also have an adaptation to ensure there is the least possible water loss ever. At the surface of the leaves, there's waxy cuticles that prevents excessive water loss when stomata are open. See, the water goes in and goes out through the stomata into the leaf cells, leaf surface. Continuing on, I will explain about the opening and closing of stomata. The opening and closing of stomata relies on is also regulated by light. As you all know, the stomata opens in the morning and closes in the night. It opens in the morning because the morning is when plants undergo photosynthesis, whereby it needs carbon dioxide, oxygen, and water. Hence why the stomata is open. So as we all know, each stomata are guarded by guard cells. Here, these two things that looks like peanuts are guard cells. How does the stomata actually close and open? Well, the stomata opens when the K plus ions, here the pink things, enters the guard cells through active transport, hence causing water to also enter the guard cells through osmosis and creating a trigger pressure at the guard cells, forcing them to open. And that's how the stomata opens. See? The trigger pressure, the guard cells are all tense due to trigger pressure, and there's a stoma here, stoma opening. So, how does the stoma close? The stoma closes when the earlier K plus ions and water molecules leaves the guard cells, uh, diminishing the earlier turgor pressure, hence causing them to close. And we will see here. We cut and close the hole. So, in addition, the addition of abscisic acid can also cause the stoma to close. That's all. Thank you. And I'll leave to Izati to continue this presentation and finish and close this also. Thank you very much. Bye.
Hi, I am Izati, and today I will explain about the transport mechanism in plant, which is organic nutrient transport. At first, we must know what is role of phloem. Phloem can transport sugar. Phloem saps. What is phloem sap? Phloem sap consists of sugar, hormone, mineral element that can dissolve in water. So the phloem sap doesn't only move upward or downward. It can move in any direction. It can travel from the source, which is the sugar origin, like all the leaf, and to the sink, which is the sugar are unloaded, like a root, that the body part of the plant that didn't do the photosynthesis process. Okay, so when we grading of tree, below the level of leaf causes bark to swell. Just above the cut, which is the sugar accumulate at the swollen tissue. So we can also did the radioactive traces studies to confirm that phloem transport organic nutrient. Okay, so let's move to the next slide, which is we can see the radioactive traces study using carbon-14. The carbon-14 labeled as carbon dioxide is supplied to the mature leaf. Radioactively labeled, sugar is soon moving from the source leaf to sink leaf and roots. As you can see, there is a picture that provided that can know which is the source and which is the sink. Okay, next we will learn about the pressure flow model of phloem transport. Sugar is actively transported from a mesophil cell, which is from the leaf, into the sieve tube cell of phloem. As you can see in the figure provided, there are active transport that using the proton pump. Active transport is dependent on an electrochemical gradient established by proton pump, and the sugar will be co-transported with the hydrogen ion that are moving down their concentration gradient. Okay, let's move to the next slide. High concentration of sugar in the sieve tubes causes water to flow in by osmosis, and the sugar solution under the massive pressure at the source is forced to move by bulk flow to the area of lower pressure at the sink-like root, and then at the root, the sugar transport out of the sieve tubes into root cell, and then the sugar is used for cellular respiration or other metabolic process, and water exit by osmosis and return to the xylem. And if you want to see the summary of this whole process, you can see in the figure provided in the cell. Okay, let's move on to the next slide, which is that we can learn about the acquiring phloem sap. We can acquiring the phloem sap by apit. Apit is a pest that we can find in the garden. The high pressure of sucrose in the phloem allows apits to insert stylet, which is the needle-like mouth part of apit, into sieve tube. And the sucrose is forced through the digestive tract, resulting in a sucrose droplet, which is honeydew, that is exerted on the rare and used by ants, who then protect the aphids. In turn, the ant that consume the honeydew will protect the aphids. And the scientists can also analyze phloem sap by cutting the aphids from its stylet left in plant tissue. So, that's all from me and my member group. I hope you can understand this chapter well, and you can ask us any question if you want. Thank you.